Okay, students, so now we are starting to learn about the carbohydrates. So this is where you can, this is the part of the chapters that we will be covering today. Okay. So why do we need to learn about carbohydrates? Okay. So carbohydrates, you see in the places around you. So for example, a common example would be, let's say, in cell membranes. You don't see the cell membranes though, but probably I would start with something we can be relating with. For example, cotton, right? So cotton is made up of cellulose. Okay, you have papers. At least some of the papers are made up of cellulose. Then you have a majority of the tissues in the plants, okay, are made up of cellulose. Cellulose has a polymer of glucose, and it's as you can see, it's a carbohydrate, right? Mm, inside the cells, or on top of the cells, where you do not really see them that well, but there is also carbohydrates present. So, for example, this is a cell surface. Okay, on top of that, you have a glycoprotein, meaning you have a carbohydrate chain linked with the protein. Okay, and there are glycolipids, where carbohydrates are covalently linked with the lipid molecules. Okay, so most commonly through phosphates. Okay, most commonly through phosphates. So this side is a carbohydrate, and this side is a lipid chain. Okay, like that. Similarly, on the proteins also, this can happen, where you can have like amino acids like serine or other side chains that can link with the carbohydrate, or it can be happening through post-translational modifications also. Okay, so what is the function of carbohydrates on the cell surface? So this provides typical cellular recognition with other entities that would like to interact with the cell. So, for example, if you know that, for example, SARS-CoV-2 virus, okay, so SARS-CoV-2 virus that has a unique glycoprotein, O-linked glycoprotein on its surface. Okay, which what it provides is that it provides its immunity against the host immune system. Okay, so that it does not really, the host immune system's antibodies do not really recognize the SARS-CoV-2 protein on top of its surface due to this O glycoprotein or linked glycoprotein. Okay, so this is part of the immune system as well. So where do you see carbohydrates? So, for example, some other areas is that, so bacteria which forms biofilms, the biofilm is predominantly formed of polysaccharide. Okay, so that is again a carbohydrate. And so if you remove the polysaccharide film, you can destroy the bacteria from the surfaces. So this biofilm formation is a major mechanism of antimicrobial resistance. Okay, so destroying biofilm, antimicrobial resistance. So destroying biofilms is one of the objectives of antimicrobial research. Okay. So some other, as I said, cellulose is polycellulose polysaccharides is a carbohydrate. Lignin is not really carbohydrate. It is present in the plant tissue in a major quantity, but it is not a carbohydrate. It's a polyphenol. But cellulose, for example, this is cellulose. So it is a carbohydrate. You can see this is a glucose units linked together. So if you can degrade and make it into ethanol, that is a major pathway for biofuel generation. Okay, so that is like plant biomass to fuel. Right, so it's a thriving research area. Okay, so looking at carbohydrates, so what do we look at? What are we? So first of all, we want to understand their structure. So here what we are looking at is a DL nomenclature. Okay, so you can see we have learned that if you have the most oxidized carbon on the top and least oxidized carbon on the bottom of a Fisher projection, if the NH2 is on the left side, it becomes an L. So in this case, an L, and H on the right side, or otherwise, if it is other way down, that would be a D. So NH2 on the right side is a D, So and this is for the L. So this nomenclature, as we know, is applicable for this carbon only. Okay, this carbon only. When we are talking about molecules like carbohydrates, so this is basically a carbohydrate molecule. We just, when we want to name the molecules with the carbohydrate or the DLL, we always talk about the last. We always talk about the penultimate carbon or the last chiral center. Okay, so this is the last chiral center on which we should be looking into for DLL nomenclature. So this particular one, for example, is a D. This particular one is a D. Okay, so we are not considering this carbons. Okay, so this they are not considered for D and L nomenclature. For DL nomenclature. Okay. So yeah, so just the general rule, let's revise. Most oxidized carbon on the top, least oxidized carbon on the bottom. Okay, and then the last carbon, penultimate carbon, whatever it is showing D and L, that is would be for the that would be the nomenclature for the molecule. So some new concepts in stereochemistry we already introduced. So first thing we want to talk about is a diastereomers. So diastereomers we already discussed and we will very quickly go over it. So it is basically where the molecule has two or more stereoisomers, okay, and two or more chiral centers, okay. So let's say that in one of his stereoisomers, not both of them got changed. Okay, so for example here, if you look at the first two molecules, you can have a look that, just a second, you can have a look that this carbon here is changed, right? So this is OH on the left side and this is OH on the right side. This carbon, this position and this position has changed. So both the positions has changed, right? So this is not a diastereomer, this is an enantiomer. So when both the stereocenters are changed, that is an enantiomers, okay? When N out of N stereocenters are reversed, okay, so that leads to an enantiomers. But let's say even up to N minus 1 stereocenters, they are changed, that leads to diastereomers. Okay, so for example, let's look at this molecule and the last molecule. Okay, so you can see this carbon, the first chiral center, the OH is on here, and on this the OH is on the other side, right? But NH2 carbon, this particular chiral carbon, the NH2 is on the right side, NH2 is still here on the right side. So this relation is a diastereomer. Okay, so this is a diastereomer. Okay, so similarly you can, I have written the answers here, so you can figure out, so which is diastereomer, which is enantiomer, and so on. Okay. Some broad introductory ideas that we want to first learn before we go ahead and jump into the sugar chemistry is that, so sugars or carbohydrates can stay in both linear and cyclic form. So for example, this is D-glucose. This could stay, and this is the linear form of D-glucose, and this is the cyclic form of D-glucose. Okay, so both can exist in nature, although we will more commonly encounter the cyclic form. Okay, so we see, for example, in the DNA, we see we usually see the ribose or deoxyribose sugar in the cyclic form. For example, here you can see this is a cyclic form. This is a cyclic form, and so on. So this is deoxyribose sugar. Okay. So one more general idea that we want to know is that the sugars can be monomeric like nucleotides or polymeric like oligonucleotides. So this, for example, is a monomeric sugar, is a single cyclic sugar unit. Okay, it can be two of them linked together like a disaccharide. Okay, so it's a disaccharide, that is two of sugars linked together. It can be a more than one sugar linked together, so that we call a polysaccharide. Okay, So one thing that we will be learning soon is the presence of the glycosidic bond. Okay, so you can see here this particular OH. So if we, this is the number one carbon. Okay, that is number one carbon. So it is already numbered here. The OH that comes out of the number one carbon, we call it the glycosidic bond. So this bond here is a glycosidic bond. This bond here is a glycosidic bond. Glycosidic bond and so on, glycosidic bond. Here in this picture, this is the glycosidic bond. This is the glycosidic bond. This is the glycosidic bond. Okay, so that is that. Okay. So now, how do we write? So basically, when we are writing structure of a monosaccharide, we have some general formats for writing. So here, if you have a carbonyl and several alcohol groups, it usually the name ends with a O's. Okay, for example, glucose, pentose, mannose, fructose, aldose, etc. And we usually write it like the most oxidized form in the top and the least oxidized at the bottom. And in the middle, we have the OH groups on either orientation. In some cases, if it's a, if it's a if it is a, if it's a ketose group, it can be the OH double bond can be also in the middle. So this is where we are talking about. So if it's an aldehyde group that is making the carbohydrate, okay, and as by nomenclature we should put it on the top. We call this aldose. So aldose coming from the name aldehyde group. Okay, if it's a ketone group, that we are calling it a ketose. Okay, so that is that. So in either case, the OH groups that form the chiral centers, for the aldose, it is right here, and for the ketose, it is here. Okay, and the carbonyl group is right here. Okay, so this structure has a little bit of problem. So how would I think I would better write it is like this. So OH on the top, okay, and then carbonyl, okay, and then we should write it as HOH and N, and we have the OH at the end. Okay, so this is a more appropriate orientation or way of writing the ketose. Okay, so this is CH2OH on the top, CH2OH on the bottom. Okay, so basically this is how. So for example, a number of sugars that are here, and you can see some of them are aldoses and ketoses, and you can see that this is the conventional way of drawing. All of them have the aldose or aldehyde group on the top, okay, and the CH2H on the bottom. So this starts with 3 carbon carbohydrates, that is glyceraldehyde, and then 4 carbon, that is erythrose and threose, then 5 carbon D-ribose, arabinose, xylose and lysose, then 6 carbon, that is allose, altrose, glucose, and there are some few others. Okay, and then we have the ketose, that is fructose, cyclose, and erythrulose. Okay, so that are, these are the ketose. So as I was saying, this is the second position has the keto group, and then the OH below, and so on. Okay, so that is that. Just to see something here, these ribose, so this is the carbohydrates linear form that is present in the RNA. Okay, once you remove the, so this is O1, 2, 3, 4 and 5, okay, 5 carbon. Okay, so this is what we say here. If you remove the position 2 OH, so this it becomes H, so that is the deoxyribose. Okay, that is what we find in the DNA. Okay, so that is that. Okay, so why all these things are named as D? Because if you look at the last carbon, the penultimate carbon, last chiral carbon at the penultimate carbon, all of them has a D configuration, right? So this is that all of them has a D configuration. So that's why these are D. Okay, not because of other positions. Okay, so that we have to be very careful about. Okay, so this is what you are just referring to dianenyl nomenclature, and that is the penultimate carbon we are talking about at this position. Okay. One more way of naming the sugar is that it is based on the D and L nomenclature and the aldose and ketose. So for example, glyceraldehyde, it's a aldose, right? So you can, and this is the D, so you can write this as D aldotriose because it's a 3 carbon backbone. So D aldotriose. So for example, here this is a 1, 2, 3. You can write this as D keto triose. D because of this carbon, and then keto and triose. Similarly, this is 1, 2, 3, 4, 5, 6. This is an aldehyde and this is a D. So D aldohexose. So this is basically glucose, but you can also write this as D aldohexose. Okay, so that is that. Yeah, so just quick question. I have this here. So what is the relationship between this particular molecule and this particular molecule? If you look at this, this has only one chiral center, and you have reversed the positions. So this is a mirror image if you put here. Okay, this OH is reversed. So this is basically a mirror image of each other. So enantiomers. Okay. Okay, so now what we want to learn how linear sugars form cyclic sugars. Okay, so that mechanism is discussed here. So here we have an aldehyde. Okay, so before understanding that, we want to understand a concept called hemiacetal or acetal formation. For ketones, it is called ketal and hemiketal and ketal formation. So let's say this is an aldehyde and this is an alcohol. Okay, so this becomes alcohol and this is an aldehyde. So what can happen is this alcohol has OH has lone pairs. This lone pairs can attack this carbon because this is an electrophilic carbon. Break this carbonyl bond and form a bond here. Okay, so this is this OR. This OR came from here, and this is the original molecule R1 over here, and this is the OH. So here what happens is this particular formation bond is called, and you can see there are two OH and OR present on this particular carbon now. Okay, earlier it was only one carbonyl molecule present. So this particular entity is called hemiacetal. Okay, then if a second alcohol attaches to it and replaces this OH, then it becomes a OR3 from this one and OR2 from this one. This is called an acetal. So to test this, let's try this out. So this is, for example, this, what do we call this, aldehyde, hemiacetal or acetal. Okay, and this particular one, what do we call this, hemiacetal or acetal? So this particular one would be called hemiacetal because this has a OH group present, and this particular one would be called acetal. So similarly for ketones, if you are forming an OH group reacting with this and forming this OR2 and the OH, this is a ketone. So here you have a H and here you have an R. Okay, this is called a hemiketal, and here we are forming a ketal. That is when one more alcohol reacts, we call it ketal. Okay, so that is that. Okay, so just had some questions here. So step A, this is what kind of reaction? This is called a substitution reaction. Okay, sorry, this is an addition reaction. Okay, step B, what kind of reaction? Step B is right here. So it substitutes this OH. So this is a substitution, and A plus B. So this is basically, and this is a substitution addition. Okay, so that is what kind of reaction these are. Okay, so using aldehydes, this hemiacetal and hemiketal formation, this is how the linear aldehydes form into cyclic forms. So here, for example, this is this particular one is a linear aldehyde. So what is happening here is that this particular OH, look at this particular OH, has a what do we say, red one, because we will be using this for forming the the ketal, sorry, hemiacetal. So what we do here, we are doing something called a 3 interchange. So 3 interchange is this bond H moved here, OH moved at this position, and CH2H moved at this position. So the purpose of doing this, as if you are rotating a bond here. Okay, as if you are rotating a bond here. So you are interchanging these groups like a clockwise version. Okay, so the purpose of doing this is that the stereochemistry does not change. So now you have put the OH in this position. What you are going to do, you are going to react this OH on this carbon. So this I have shown here with a folded form. This is the terminal OH, and this will react with the C double bond O and form the bond here. So this bond formation, this opens it up and forms a hemiacetal. Why acetal? Because originally it was an aldehyde, and why hemi? Because it still has a it still has a OH present here. Okay, so that is called a hemiacetal. So in this cyclic form, this particular one is in this cyclic form it is called alpha D glucopyranose. So let's understand why that is. The reason why it is alpha, because this OH group right here is down. Okay, so this is pointing down, and D because it was originally a D. Why gluco? Because it was originally a glucose. Pyranose, because pyran means it's a six-membered ring. So six-membered ring oxygen, six-membered oxygen rings are called pyran. Okay, and five-membered rings are called furan. Okay, so that's a pyranose. Okay, if during this reaction the OH could have formed up, then it would be called a beta D glucopyranose. So everything is same. Beta means this OH is pointing up. Okay, so that's a beta D glucopyranose. Okay, so that is that. So so I will stop it here, and this will take up from this particular slide from now onwards.